Hello, I'm Akram Yazdani, and I'm here. I'm currently at University of North Carolina at Chapel Hill, but I'm joining the University of Texas at Houston as an assistant professor. And I'm happy to talk about one of my works on immune response-related gene regulatory pathway in colorectal cancer. Uh, it worth noting that colorectal cancer is the third leading cause of ca- mm, cancer-related deaths in the United States, and there are targeted therapies for metastatic colorectal cancer. But although they have improved progression-free survival, but we observe that there are large variation in clinical outcomes among patients. So it means that there are some unknown mechanisms that are affected by inhibition of these targeted therapeutic pathways. So understanding about the mechanism, these unknown mechanism can help to improve the efficacy and a response to treatment, and also, of course, reducing the toxicity of the treatment. So when we are talking about mechanistic understanding, we need system approaches to answer this question. So we introduce an integrative system approach for our study of cetuximab and bevacizumab that are targeted therapies for uh, KRAS wild type advanced and metastatic colorectal cancer. The proposed approach is incorporating the information from different types of data, from the tumor tissue RNA-seq data, clinical data, and also germline genotype data. Integrating this information in the analysis of analysis that integrating conventional approaches, Mendelian randomization, and causal networks. So what this approach offers is to find the genes that are causally associated with overall survival, and also mediator and confounders. And finally, we can find the gene regulatory pathways that are related to overall survival. So briefly, I want to mention that when we are using integrative approach, we need a study design. We need to develop an approach based on what we have in our hands, not developing an approach and then see how we can use for performing that. So it's very important. As you can see, we have different types of data. The overlap of the which samples we have are different, so we have to define how to use those and which approach to integrate. So for finding the genes that are causally associated with overall survival, we propose a Mendelian randomization approach, which is based on mm, uh, considering that genetic variation can be used as instrumental variable. Instrumental variable should satisfy three assumptions that you can see in this diagram, which is known as causal diagram. So using this causal diagram, we reduce the dimensionality of search space using conventional approaches like CCQTL analysis. So the main steps and essential step in Mendelian randomization is to test the assumption. That is, the IVs are valid, or if we have the pleiotropic effect or not. So in the first stage of analysis, we test the validity of IV by predicting the expression level of the gene expression gene, and then assessing that if this prediction is still associated with overall survival or not. If this is still associated, it means that it can be valid and a strong IV to use for causal interpretation. The second step is pleiotropy effect. So it's very important to not to have a pleiotropy, because if we have pleiotropy, we cannot talk about causality. So for assessing that, we are doing some GWAS analysis first to see which SNPs or IVs are potentially can have a pleiotropic effect. So in this case, we need to reduce the GWAS threshold because GWAS threshold is very strict for this type of analysis, and we put ourselves in the risk of missing pleiotropic effect. So after selecting these SNPs, we perform a additive model that has IVs and genes at the same time in the model. If the only IVs is significant, it means that we have a pleiotropy effect. But if the genes still is significant, uh, its relationship with the overall survival, then we have the genes that causally associated with overall survival. So in each steps, we try to do replication. Using the data we had, we decided to use the data that for the samples that we have only genotype data for the replication. But is it working properly? Since we have the only genotype data, we use the two sample MR analysis. And we replicate the result. So for finding a gene, the next step, we are trying to find gene regulatory pathways related to overall survival. We build a causal network, which is an augmentation of Bayesian network and principle of Mendelian randomization. I should emphasize that the focus of causal network is not just interconnectivity between genes. It's more focusing on finding directionality, that which gene are regulating the other genes. So this one of our approach in this context was recognized at 2015 causal inference. And but here we adapted because the data was different and we need to adapt and modify that approach in order to integrate background knowledge and the result of MR analysis into the study. So we use two different criteria in order to build the network to see how sparse the network can go. We use the Hamming distance, and then we decide of the threshold how to stop that, which is in the paper, more details. And also we build the network, dense network, and the overlap between these two were the one that we selected as the called true interconnectivity and at this stage. So replication of a network is usually is very difficult, but we used another arm of a study that was prematurely stopped because of low efficacy for the replication. So when we have this type of causal network, we have directionality. We can define which gene is a confounder, which gene is mediator, respect to the overall survival. Then we can reassess the relationship between gene and overall survival using a structural ecology modeling. So here there are three examples I put as we call it pathway, but you can call it module or a subnetwork that have been replicated and have the genes related to overall survival. And also we have two different treatments. So we compare the effect of each genes on overall survival for each treatment that you can see in this bar plot. Since we did this approach and we try to replicate all the results, it was not as challenging as we expected for the network analysis to interpret the result. We did we cluster all the genes in this three subnetwork, as you can see, based on 11 immune signatures. And as a result, for example, one of them, if you see, all the genes in these pathways are related to wound healing. So it makes sense, and we can easily interpret what this module or subnetwork is for. is doing and what's the function. So as a conclusion, I would like to say that uncovering the interconnectivity between genes through the integrative system approach based on Mendelian randomization leads to a better understanding of the therapeutic effect on overall survival and finding reproducible and interpretable results. Finding novel biomarker that may lead to personalized therapies, because we were comparing the different treatments that could help to decide about that, which treatment is better for which patient. And also, I always say that we need a study design for integrative approach to have better result for the analysis. And I would like to acknowledge all my collaborators and co-authors in this work, and also Alliance, Genentech, Pfizer, and Sanofi for their financial support and preparation of the data. And also thank you for attention, and if there is any question, I will be happy to take. Yes.
Have you considered sort of the interaction between genes that are chosen as
Instrumental variable, by nature, we use just genetic variation, because we know that the direction of genetic variation always is to the gene expression and vice versa. That is the main assumption we have to make. So we don't use the gene expression as instrumental variable. Yeah. No, because we are not sure. Because if you look at the causal diagram, we are defining this directionality. If direction is not working, we are not sure about that, so we cannot talk about causality. Yeah.